Practice 5. Polymerase Chain Reaction (PCR).
Introduction. The polymerase chain reaction, or PCR, is a very used technique in the genetic sciences. This technique allows the exponential multiplication of nucleic acids, meaning either DNA or RNA. In the most general way, they both work in the same way. However, the sole difference between them is that when working with RNA, it needs to be first transformed to DNA. The mechanics of it are taken from the knowledge there is regarding DNA replication. This is, for it uses, like in the replication of the lagging strand of DNA, a set of primers so that the polymerase can act in the correct direction. Just as it is used to create many copies of DNA in general, it may also be used to multiply a very specific site at the genome, like a gene. In this case, the genes that are planned to be analyzed are the ZFY genes, which can tell the difference between male or female.
First of all, the selected gene to make the replication in PCR was CFY. The methodology employed on PCR followed those steps. In first instance, was necessary to unfreeze the DNA sample, and then was put on ice. Was necessary to make a master mix which includes some components. Only the following volume values are in microliters. The buffer was with 15, DNTP 3, primer forward 3, primer reverse 3, DNA polymerase 0.6, and finally molecular biology water 103.5. The total volume of the master mix was 143.1 microliters. This master mix was divided on 3 microtubes. Each of them was 47.5 microliters of the mix. Apart from the master mix, in each microtube was added other components. In the first microtube was the problem sample, with 2.5 microliters of the DNA sample. In the second one was the positive control with 1 microliter of PCR product and 1.5 microliter of molecular biology water. And in the third one was the negative control with 2.5 microliters of molecular biology water. The thermocycle had the following indication: 44 degrees Celsius, 5 minutes for the neutralization, and 30 cycles of 3 steps. 94 Celsius grade for 30 seconds, 59 grades Celsius for 30 seconds for alignment, and 68 grades Celsius 30 seconds for amplification. In the final steps, 68 grades Celsius for 5 minutes and then hold in 4 grades Celsius on temperature. Finally, to visualize the PCR product was necessary to do an electrophoresis using 1% of agarose and a buffer TBE 0.5. The specifications of electrophoresis were 110 volts for 40 minutes.
Results. In the first well, there is the DNA ladder, which is oxygen 1 kV. In the second well, there are 3 lines, which are PCR product. In the third well, there are 3 lines, which are the positive control. And for the last, in the fourth well, there are 2 lines, which are the negative control. In the image of the electrophoresis, there is a DNA ladder. In the analysis, the normal oxygen DNA ladder are 13 lines, but in the image are only 12. It could be because the agarose percentage is more than the necessary, the voltage was lower, or the time of the electrophoresis was lower. In other hand, in the second well was the PCR product, and it seems a specific product, and according to the oxygen properties, the size of the PCR product could be between 400 and 500 base pairs. After that, the estimated size is 490 base pairs. The light marks under the PCR product could be DNTP, or primers, which was not used during the amplified. In the third well is a positive control, which contains just DNA without amplification, and also it seems with a smaller size as a PCR product, and also at the bottom are the NTP, which was not used. In the fourth well, there is only water for the negative control.
In conclusion, the results obtained by the experiment were satisfactory. The correct amplification of the genes was observed in the gel. However, there were observed several factors that interfered with the correct resolution of the sample. These abnormalities may be originated by a change in the concentration of agarose used to make the gel. A more careful elaboration is recommended, and it is strongly recommended to avoid variations in the voltage applied.